Mr. Tazbaz starts, just for the panel to know, we are now going to have the first 5 presentations of our agenda, and we will then be able to ask questions after we have the next 5 panel presentations. Mr. Tazbaz, thank you.
Thank you. Good morning, everyone. I first want to extend my heartfelt gratitude for all of you for being here and dedicating your time, your knowledge, and expertise to these very, very critical discussions. I also want to thank the FDA team for working tirelessly to making this event happen. It was difficult, but it's going to be very well worth it, and we're very excited about the discussions that are going to be taking place today. The Digital Health Advisory Committee has attracted significant interest from the entire healthcare community. We believe this is a reflection of the growing role digital health technologies will play in advancing patient care and overall health of our entire population. As the FDA's first advisory committee focused on cross-cutting digital health issues, we hope DHAC, as we've been calling it, will guide us on matters that affect the wide spectrum of medical device products. Today, we are here to address one of the most transformative developments in healthcare, generative AI, or as we've been referring to as GenAI. We hope to explore and identify unique regulatory considerations necessary to ensure that safety and effectiveness of these rapidly evolving technologies are maintained throughout their life cycle. Generative AI brings incredible opportunities to enhance patient care, but also introduces new regulatory challenges and oversight, and operational oversight. As it's been highly covered, generative AI has the potential to reshape healthcare by generating contextually relevant outputs from vast amounts of data. However, unlike traditional AI models that predict or classify data, GenAI produces new outputs, which can introduce additional layers of regulatory complexity for us. This is especially relevant when these outputs could directly impact the safety and effectiveness of medical devices. To guide us throughout the discussions, we would like to take a total product lifecycle approach, as Michelle mentioned. The lifecycle perspective allows us to consider safety and performance from initial design and development all the way through the post-market monitoring. Throughout the next 2 days, we will dive into critical questions surrounding generative AI, including pre-market evaluation, performance metrics, and effective post-market strategies. Additionally, we will discuss the complexities of monitoring generative AI models across multiple sites, as we need to ensure that they perform consistently while managing potential regional biases and data variations. Our agenda is designed to give structure to these important discussions, beginning with an examination of information needed to evaluate generative AI-enabled devices, followed by in-depth sessions on risk management and post-market performance monitoring strategies, which is a very important part of the equation of when these things are deployed into real healthcare settings. As we move forward, I'm confident that the insights and ideas shared here will shape the FDA's approach to balancing innovation with safety and ensuring that the patients and healthcare professionals can trust these powerful new tools. I would like to say thank you once again for your participation and engagement. I look forward to the rich discussions ahead and the actionable steps we will identify here together. In a moment, you will also hear from Dr. Aldo Badano on some regulatory science challenges that FDA faces for evaluating generative AI applications in medical devices. And tomorrow, FDA's Jessica Paulson will provide an overview of approaches for managing changes for AI-enabled devices. Thank you, everyone.
Thank you, Mr. Tazbaz. We will now proceed with Dr. Aldo Badano's presentation. Dr. Badano, as mentioned, is Director of the Division of Imaging, Diagnostics, and Software Reliability in the Offices of Science and Engineering Laboratories at CDRH FDA. Dr. Badano.
Good morning. Thank you for the introduction, and it's an honor to speak to the distinguished members of the panel and the audience. Let me start by saying a few words about our office. OCEL in CDRH, Office of Science and Engineering Labs, is an ISO 9001 certified research organization dedicated to promoting innovation for the development of new life science medical devices. OCEL is organized into about 20 program areas, and AI/ML is one of the largest programs that we have. OCEL outputs are regulatory science tools, which are innovative open source tools for assessing the safety and effectiveness of emerging technology. Innovators can use these tools voluntarily into all stages of device development. Our regulatory science tools have been cited in over 1,000 pre-market applications to the agency, over 100 product codes. If you would like to learn more, there's some information about how to reach our regulatory science tools catalog with a website and the QR code. GenAI-enabled devices in healthcare have a wide range of uses. Here's a table taken from the literature, an article by Mexo and Topol, which outlines some of the applications and how the community thinking about GenAI in the medical space. Some of these products are clearly non-medical devices, but there's a lot of excitement in terms of the potential performance and productivity gains. However, the same characteristics that make these devices and products have so much potential are the characteristics that might pose challenges for certain uses. So what are the regulatory science challenges that this technology brings about? Here's a preliminary list. I'm not going to go into detail in all of these, we don't have the time, but many of these challenges might exist in other technologies but are exacerbated in AI applications, particularly in GenAI. So difficulty in defining scope of the product's intended use, some of the parts of the model, like foundation models, not being in the provenance of the manufacturers of the device, oversight of adaptive systems, and hallucinations, which are a new kind of error that has been introduced in recent years by deep learning and other technologies that bring about a new trade-off between stability and accuracy, leading to concerns about reliability and trustworthiness. There's also issues about data needs, including diversity, monitoring performance in the real world, including bias, transparency to users. So let's narrow it down a little bit with one example use case. In this use case, a GenAI-enabled radiology device can produce a report when prompted with a medical image, as you see in the left in the input side. The reference for this device might be a clinician-generated report. So what strategies do we have at hand to evaluate these new technologies? In recent years or months, I would say, three distinct classes of evaluation have emerged from the scientific literature. Benchmarking with the standardized reference data sets, expert evaluation, including a holistic evaluation of the AI-human interaction, and model-based evaluations, which include automated testing. In the next slides, I'll describe a little bit the pros and cons of each one of these approaches. Benchmarking. Benchmarking is the evaluation of models on external testing data sets with predetermined evaluation metrics. This approach is practical, allows for head-to-head comparisons of models on same data and with same metrics, and can assess models at a large scale. However, benchmarks are limited in models and in tasks or applications. And there is this risk of that the models can train to the test, which is an overfitting technique for optimizing performance on a given data set at the expense of performance for other data distributions, for instance, like the ones you see in a different clinical setting. On the right, you can see one of the many leaderboards available in open source formats. Expert evaluation. Expert evaluation refers to using experts to establish the reference standard for a given task. For example, experts may determine if an error from GenAI will cause significant patient harm. Note that in the chart, experts can identify errors that might pass the tests of a benchmark concerned only with text similarity metrics. For instance, right versus left, which is location of disease, fusion versus effusion. Expert evaluation is adaptable to a wide range of tasks with direct clinical relevancy. However, expert evaluation is resource intensive and burdensome and must consider the subjective nature of the readers and the highly variable effects. And finally, model-based evaluation. This is about evaluating models with other models, using a model-based approach that must have human oversight. Let me bring up the chart. The advantages are clear. This augments the human evaluation, is scalable. However, the disadvantages include the fact that the validation needs to be robust. And there is a potential risk of intermodal leakage, which means models in the device and in the evaluator can be correlated, therefore generating risks of the results. In our CDRH labs, we're actively investigating a model-based evaluator for factual accuracy of and GenAI-generated radiologic impression. A series of binary questions are used to determine the rates of different types of errors in the generated report. We're also working on performance metrics and associated statistical analysis plans. I hope that you'll stay tuned for updates in the coming months. Let me summarize. For some GenAI, known evaluation strategies still may apply. However, new evaluation methodologies and new performance metrics may need to be developed for some other types of GenAI. Overall, device performance evaluation requirements are governed by the intended use and the associated risk for the device. And finally, particularly in the case of GenAI-enabled devices, evidence may include pre- and post-market elements, as the technology is meant to update often based on the availability of models that are trained on more and more data, and the emergence of novel architectures and GenAI innovations. In summary, rigorous evaluation of GenAI-enabled devices is necessary as a rapidly emerging technology developing a least burdensome approach to ensure safety and effectiveness will require partnerships like the one we're seeing here in this room and commitments to advancing regulatory science. Thank you for your attention.
Thank you, Dr. Badano. Next, we're going to hear a stakeholder perspective from Dr. Faisal Mahmood. Dr. Mahmood is Associate Professor at Harvard University Brigham Women's Hospital. And just a reminder to the panel that when we are done with these, we'll take a short break, and then we can ask questions of the panelists. That includes Mr. Tazbaz and Dr. Badano, so just save your questions for them as well as our next three speakers.
Okay, thank you for having me today. So, so I'll be talking a little bit about some of the work that my group has done over the years in computational pathology and how how it has led into generative AI and what we are currently focused on. It's just a quick outline for the talk. I'll be talking about some of our older work and how it feeds into the generative AI related work that we have done more recently. So the problem that we're trying to target is to analyze pathology images. Pathology images are hierarchical, they're large, they're very large gigapixel images that result from the digitization of glass slides. And the goal of our field is really to go from these images to everything in the red box here. This involves early diagnosis, prognosis, prediction of response to treatment, outcome prediction, patient stratification, and some form of biomarker discovery. So I'll start by talking about some of the sort of quote-unquote historical work, even though it's quite recent. So this is some work that we did back in 2021. This allowed us to use these large whole slide images and slide-level labels that existed in pathology reports while still being data efficient, so without requiring any pixel-level annotations. And this was published back in 2021, and we made the code and the models, everything publicly available for the community to use. And these tools have been extensively used and have basically been applied to every major disease organ system. And we applied it to a number of different applications, including for cancers of unknown primary, where we tried to find the origin of the tumor. It's a very common and difficult problem in pathology. And we posed this as being an 18-class supervised classification problem using whole slide images and labels that were taken from a pathology report. And overall, we found that this approach works quite well. Internal and external test sets. We did a lot of evaluation, assessing the variability. And then more recently, we have tried to combine histology images with molecular data to see if we can improve outcomes, and have shown that this is also possible. We're also interested in integrating histology with molecular information to improve outcomes in general. And have shown in this study from back in 2022 that you can improve patient outcome stratification into distinct risk groups, but then also go back and identify biomarkers that would be relevant to the diagnosis, but also would correlate with response and resistance to specific therapeutic agents. It's a bit slow. This is another example for endomyocardial biopsy assessments. So our approach has been that we can train these supervised models, but then targeting problems that are difficult to solve in pathology. So hence, it would sort of encourage machine learning adoption, because these problems are difficult to begin with, like cancer of unknown primary or endomyocardial biopsy assessment, where there is large-scale intra and intra-observer variability. But the purpose of showing all of these examples is that they all follow this very conventional approach that's commonly used in computational pathology, where we have whole slide images that are digitized. We patch them because they're very large. We extract relevant features, or in this case, they're exhaustively extracted from a pre-trained encoder, leading to aggregation and eventually a prediction. This pipeline has been innovated on in various components over the years, both by the clinical community as well as in terms of how the data is used, and by the machine learning community in how we feature extract and aggregate. But over the years, we have found that the feature extraction component is the most important component, and that's what leads us into foundation models or self-supervised models. In the examples I showed earlier, we were using a model that was trained just on a ResNet that was trained on real-world images from the ImageNet database and is used for feature extraction. But more recently, we've seen that self-supervised models perform significantly better. So motivating foundation models for pathology. So foundation models are generic models that are capable of rich feature extraction. And the particular use that we have here is that by having richer feature extraction, we can apply this to situations where we just don't have enough data, very, very common in pathology, for example, for rare diseases, clinical trials, and they're ideal for multitask, multi-tissue kind of problems, and they're not necessarily meant to replace end-to-end trained supervised models. So this thinking was sort of what led to these two studies. That was from March this year, where we have two foundation models. The first one, UNI, is a self-supervised model that can train, that's trained in a self-supervised manner on lots and lots of pathology data to extract rich feature representations from those pathology images. And Conch is a vision language model, which further enhances this by contrasting with text. And this is in line with how the machine learning community is thinking about this, because they've shown that contrasting with additional modalities leads to better improved feature representation for both images and for text. So we collected a large data set of about 100,000 whole slide images. This came from a number of different hospitals, including our own, and it's diverse. It was maximized for diversity, and this is also in line with how the machine learning community has found that diversity of data is much more important than the quantity of data in building these large self-supervised models. And we used the DynoV2 framework from Meta to train these models, and it was assessed against a number of different benchmarks, both conventional and pathology related, and was applied to 33 different downstream tasks. The radar plot is not the best way to show this, but it does capture all the different tasks that this was assessed on, including for whole slide level classification, as well as for region level or small scale classification, and in particular for few shot classification, where we attempt to train these models with very, very few data points, hence assessing its efficacy on situations where you would not have enough data available, for example, for rare diseases, clinical trials, and so forth. We also wanted to see how far, I'm sorry, the colors on this slide did not show up as they originally were, but We also wanted to see how far can we push this. Can we have a single model that can cater to a really large, difficult classification problem of 108 different cancer types in the entire oncotree classification system? And the goal here is not to reach clinical utility. It would still take some time, more data potentially to get there, but to really assess that how rich the feature representations get if we use this form of self-supervised learning. In parallel, we were also working on contrasting images with text, and in this case we use 1.1 million image caption pairs that come from the PubMed open access database. We could not use all the data in PubMed, only that was publicly funded and is within the open access database with an appropriate license. But contrasting images with text is a very common approach used by the computer vision community to enhance feature representations for both images and for text. This is a distribution of all the data that was used in this particular case. And we assessed this in a number of different scenarios, including for zero-shot classification, where the goal is to really assess the feature representation and not really achieve clinical utility. But also for few-shot classification, we're showing that fewer data points can lead to better performance in a variety of different disease models, both rare and common. These models were made publicly available, and we essentially did this to comply with NIH guidelines and what the requirements were from the journals where we were publishing this. But the models were extensively downloaded and have been used all over the world for a host of different applications, or for every major organ system, all the way up to forensics, so things that we never originally thought of. We didn't even know that this many people were working in computational pathology. And they have also been assessed independently by a number of different groups. This is an evaluation from a group in Dresden, where they have shown how these models work against a number of other approaches, self-supervised and otherwise, for a host of different tasks, including diagnostic tasks, as well as predicting what's commonly referred to as non-human identifiable features, like predicting molecular markers and other alterations directly from histology images, and just prognosticating directly from histology images. Here's another assessment that was done at Mount Sinai and at the Memorial Sloan Kettering Cancer Center using our foundation models, and how well it fares for some of these very difficult pan-cancer predictive tasks. Since these models came out, we have further expanded them to a whole slide level. So the models I showed so far work at a region level, extracting rich feature representations from regions. We have since expanded this to extracting a single feature vector that corresponds to the whole slide, and we've done that on an image level, and then it's further contrasted with generated captions that come from our generative model that I'll talk about in a second, but also from the pathology report. So contrasting both with a generated caption and a pathology report. And we find that this form of feature extraction, where we have a single feature vector for the whole slide, essentially enables a lot of different things that we can do downstream for incredibly difficult tasks, and for common clinical tasks. For common clinical tasks, we can get clinical grade performance, and for more difficult tasks, we're further trying to improve the model to sort of get there. But I think what most people get very excited by is that its ability to perform on few-shot examples in situations where diagnosing is incredibly difficult. Average diagnosis for rare disease is between 6 to 8 months, and it really is a challenging problem, where we can use these rich feature representations to find similar cases, but also train with very, very few cases. And then, of course, because it has a text head, we're able to generate these reports. We compare it with pathologist given reports, and we've done quite some analysis on how good the model is for report synthesis, both at the level of the slide for the patient, what happens when we include electronic medical records, and so forth. So the story so far is that we started with lots of supervised models. We had self-supervised models or foundation models that relied on images themselves, and then we contrasted those images with text to improve the image feature representation. So there are a lot of other modalities that we have in pathology and in healthcare in general that can be used to contrast data to further improve feature representation for each one of these individual modalities. So we contrast in this example. This is an article from earlier this year. We contrast pathology images with immunohistochemistry, and by contrasting, we further improve the feature representations with just a single model that can extract rich features, and then it's applied downstream to a variety of different tasks, including IHC quantification. So the first time showing that you don't need to have pixel level annotations for this very difficult task of quantifying IHCs, and it could be just this model trained on lots of IHC data that can cater to all of immunohistochemistry scoring, as well as downstream applying it to survival and so forth. And if this is true that we can contrast with IHC, an obvious next step would be that can we also contrast with transcriptomics? So in this smaller example that was published earlier this year in CVPR, we showed that we can do this and it can substantially improve few-shot performance for detecting rare diseases. But more recently, we have expanded this to a very large-scale study where we are using all the molecular data from the Brigham and MGH that's been collected over the years and correlated with histology images. And by contrasting with our NGS data, with transcriptomic data, and with histology data, we're further able to improve feature representations, and these are a variety of different downstream tasks that it was applied to, but just to draw your attention, it was able to improve performance for these incredibly difficult tasks for predicting treatment response, so often for which we have very few samples available, because at a tertiary medical center like ours, it's often difficult to get pre-treatment biopsies because a lot of patients get referred rather than the hospital being their first instance. But given the data is so small and the downstream treatment efficacy is still low for a variety of different therapeutics, improving performance by using image-based biomarkers is of a lot of interest to drug companies and other stakeholders. So this is quite exciting for us to see that for the first time we can just boost performance by using these contrastive foundation models. So what do we do after we have all these foundation models? We can, of course, train lots and lots of supervised models by using these self-supervised rich features. But what was most interesting to us is that we can also use them in a generative setting where we can have a singular model that can cater to all of human pathology. And this was the biggest undertaking that my group had done. So our entire group was involved in collecting the data for this, because the data required is not collected essentially clinically. So our goal is to build a singular multimodal large language model that can cater to all of human pathology. And why we believe this is possible is because there are an increasing number of large language models and multimodal large language models that cater to very large amount of information. Like OpenAI is trying to build a single model that can cater to the world's information, and human pathology knowledge is much more constrained and specific. So if the first is true, we should be able to build a model that caters to just all of human pathology. And what we need to do this is that we need rich feature representation that can come from foundation models or self-supervised models. We need a large instruction data set of images, text, and corresponding responses, and we also need robust evaluation. So the challenge we face is that we have these hierarchical large pathology images, but the annotations we have corresponding these images are at the level of the slide and not at a region level, whereas an effective chatbot for pathology or a co-pilot for pathology or a singular model that can cater to all of pathology will have to operate at all of these levels. So the single biggest challenge is that fine-grained understanding of pathology regions and cellular level information leads to slide-level diagnosis, but that fine-grained information does not reside in pathology reports. So this required a large amount of manual data collection, which we had from multiple collaborators across 6-7 different institutions, and the data collected also needs to be incredibly diverse and should cater to all known entities within, of course, neoplastic, infectious, inflammatory, or otherwise, that would be added. So in this initial version that was published in June of this year, we had about 999,000 question-answer turns corresponding images, questions, and answers that were used to train this model. So the image features came from the foundation models that we had developed earlier, and then it was fine-tuned on this large 1 million instruction data set. And then this led to PathChat, which is a generative AI tool that can cater to all of human pathology. It catered to a smaller subset back when the article was published, but now we have expanded this to about 13 million question-answer turns and have really extended the data substantially, with a lot of institutions volunteering data just by virtue of the fact that this was this made a bit of noise in the community. But you can ask basic questions like what am I looking at here? So it could be a very good training tool. But what we're most excited about is that it can basically work at a whole slide level, where it can generate the report, and by the time a pathologist is looking at it, they would have the entire trajectory of the diagnosis worked out and a report already generated. Another thing that we're very excited about is that it can do, it can ingest multiple images within the same context, which means that if by looking at an image, a pathologist decides that additional tests would need to be ordered, those tests could automatically be ordered, and once those images from, for example, immunohistochemistry are received, they can be ingested into the same context, and a report would already be generated. So in this example here, the report is being generated. Of course, a big utility. We had a lot of interest from a variety of different foundations who would like to use this tool in low resource settings, because there are very limited pathologists in resource limited settings, and it's the norm to send or ship out slides for a diagnosis to be made in resource rich settings. However, that's sort of prone to a lot of delays, and it's been shown that such cases, depending on where they're sent and how much data, how much tissue material is available, also have a lower accurate diagnosis rate. So in this case, the user is just taking an image with a cell phone coupled to a microscope. It's a very common approach used in lower source settings, and people take these images and send them to their colleagues in resource rich settings and trying to see that how well this worked. We did a lot of domain adaptation analysis, trying to see that these models that are trained largely on whole slide scanners from a variety of different vendors, how they would adapt to image generation in this form, and how the resolution would vary, and then went back to the drawing board and improved our training pipelines to cater to some of those variabilities that would improve domain adaptation. Another example, and something that a lot of people are very excited by, is our ability to use PathChat by just a microscope coupled to a camera. So only about 4% of diagnosis in pathology diagnosis in the U.S. is digital, and or 90% of the pathologists still use a glass slide under a microscope. Most microscopes in the U.S. have a camera attached to them. So in this case, the user is asking that this is a lymph node mass, and what's the likely diagnosis? And the chatbot says that, well, it's likely a melanoma. And the user can ask that what IHCs or additional ancillary tests could be used to confirm this. And we have a number of different possibilities. And once those tests are in, you can take an image of the IHC, and it would ingest it within the same context. And this capability is possible because during the training, we mapped out trajectory for over 100,000 cases. Of course, we had lots of one-shot training with lots of images, questions, and answers, but also we mapped out entire trajectory of cases where we start with an image, additional tests are ordered, and basically the entire diagnostic trajectory of the entire case, and use that as the training data. And we are further assessing this capability on a large data set that's collected across multiple institutions. So the most important component in building this, of course, building this took about 3 years, but in building this, the most important component for us is the assessment and evaluation, and how the evaluation needs to evolve over time as this chatbot essentially gets better. So initially, this was largely based on the qualitative analysis is based on multiple choice questions. So we basically took inspiration from 2 different domains, inspiration from how generative machine learning is evaluated for conventional, like non-healthcare related machine learning, and how large models that cater to multiple diseases have been evaluated on the healthcare side, like NGS assays and so forth, and brought the 2 together. So in this particular case, we're looking at lots of multiple choice questions and how the model fares against other generative models that are not specific to healthcare, or are specific to healthcare but not specific to pathology. But I think because this is generative AI and it gives open-ended responses, it's very important for us to assess how well those responses are given and recorded. So in this particular case, we had a panel of 7 board-certified pathologists who assessed the entire trajectory of conversation and each response generated by the chatbot and compared it against other generative models. So we collected both quantitative responses, whether the response was correct or not, the binary response, but also a more qualitative response, that how well does this does in comparison to other models. If we go and look at how other large language models are often assessed, it's often done in a competitive setting where multiple models are assessed against often a human. And we found that the models do quite well for their intended use, for diagnosis and for describing morphologic descriptions, but don't do very well correlating back to clinical, or just marginal decrease in performance, or for example, ChatGPT or GPT-4V or GPT-4o. And the reason could be, we don't know, of course, because we don't know what GPT-4o is trained on, but likely OpenAI is training it on all of medical literature, and correlating back to clinical might be much more easier in their case. We're improving the capabilities in terms of the ancillary testing, and this is what was done for the article, but we have substantially expanded this to about 11,000 cases versus 260 earlier, and have also expanded the number of pathologists who are evaluating this as we continuously improve this model. So now that we have the generative AI model, a next step is obviously to look towards agents. So we started with supervised models, and then we built self-supervised models for rich feature extraction, and we use those self-supervised models for a generative AI tool. And the next step is to see if we can build an agent. So AI agents can essentially do things for you. In our case, we do a lot of biomedical data analysis. We want to see that can we replicate what essentially we're doing every day, and could be continuous discovery agents. So I'd just like to show this one example that we have, where the user is saying that here's my data set, and I have a cohort of responders versus non-responders. Can you go train a model that would predict what can you go train a model to predict responders versus non-responders? So the model would essentially make a plan. So this plan is essentially coming from the generative machine learning component, where you would segment the tissue, extract the features, train the models, and a mechanism of evaluating this. But then it can make use of existing code and patch it together with some additional code that might need to be written. So in this case, it uses PathChat for morphologic description. It uses GPT-4o for writing the additional code that might be needed, and a host of different libraries that are out there to look at to essentially assess. To essentially conduct all of these independent tasks. So once the model is trained, we can ask additional questions. This essentially enables discovery from people who would need a clinical or a computational collaborator to run some of these experiments. And then the other benefit of this is that it can be crawling through large amounts of data, essentially doing what we do with lots and lots of pathology data manually in a pathology department. So throughout the history of pathology, the way new morphologic diseases are identified, morphologic entities that correlate with disease or outcome are identified, is that someone goes in, looks at lots and lots of different cases, and says that this similar morphology across cases correlates with outcome. In this case, it's doing this essentially on its own, and is using PathChat to generate a report. So this is quite exciting, and we have this being tested at everything we are scanning at the Brigham and Women's Hospital. So towards the end, I just want to touch upon bias and fairness in computational pathology. So this is a topic that we are very, very interested in, because we want to make sure that the models that we develop have the broadest reach. And as we go from more specific models to more generalized models, we want to make sure that their adaptability across is not just assessed across a variety of different hospitals and scanners, but is also assessed within protected subgroups, patients of different demographics. So in this particular case, we're using very commonly used data, for example, the TCGA and other public data sets that have been used extensively in computational pathology literature to train these models, and then we're adapting those to data that came from MGH and the Brigham. But we stratified that data based on race, whether the patients have had insurance or not, income status that was extracted from the postcode, and a number of different other variables. This was a study from earlier this year, And we basically want to assess that does any of these shifts that are commonly known, whether it's acquisition shift or just demographic shift, leads to any bias and disparities. And we know that acquisition shift, for example, here we're showing 3 different examples of the same slide being scanned from a different scanner, or in the case of radiology slide, the same patient being scanned at 2 different hospitals. And these cohorts that are quite often used for training do have large disparities in where the data was collected and what the demographic makeup looks like. So our sort of approach was that we would basically look at every component of the computational pathology pipeline, where we train these models in a weekly supervised manner, and assess for every modeling choice and the impact that it would have on these disparities. This includes from different pre-processing steps to the kind of features that are extracted, to the aggregation profile, and using commonly used techniques for bias mitigation, for example, using contrastive approaches, or other modeling choices that are known for bias mitigation. And overall, we found that the feature extractor is the most important component in improving fairness for these models, and this was assessed for basically every common modeling choice in computational pathology. So I'll stop here in the interest of time, and I want to leave you with this interesting poem that Judith Previtt, who's a real pioneer in analyzing microscopy images using computational tools, wrote. She was doing some very exciting work in the 1960s and in 1970s at the NIH, and then at the University of Pennsylvania. She writes something that I think is very relevant today. So optical illusions can deceive the subjective eye, but objective measurements and algorithms are assumed not to lie. It's often said that medicine could use such objectivity, and thought that this justifies machine intelligence activity. Artificial intelligence is another craze that uses computers to cope with the diagnostic maze. Though the criteria for intelligence has never been resolved, paper after paper claims that the problem has already been solved. So I think it's very important to keep in mind that there's still several challenges that we need to go through, and they're mostly related to evaluation and assessment of these very large models. So I'll stop here, and thank you so much.
Dr. Mahmood, thank you. Thank you for all the work that your lab does in this area, and thank you for being so humble about the amount of work you're doing, but the importance of getting it right for clinical care. So next we have Parminder Bhatia, who is the Chief AI Officer at GE Healthcare.
Good morning, everyone. This is Parminder Bhatia. I'm Chief AI Officer at GE Healthcare. I'm super excited here to talk about generative AI and its potential to transform healthcare. But just as important, we'll also talk about some of the technical guidelines and methods to manage the risk associated with some of this technology. Firstly, before we get started, I would like to thank FDA for bringing this committee together to discuss this highly relevant topic that has the potential to improve the quality of care for the patients, as well as reduce the cognitive overload for the providers. Just like FDA led way path for medical devices powered by deep learning to be safe and effective, they'll have a critical component in ensuring GenAI is used in a safe and effective way as well. So today, at the outline, we'll be talking about 3 major components. We'll start by diving deep into generative AI and foundation models, what they are, why they matter, how are they different from traditional AI models. Then we'll talk about their healthcare impact, where we'll talk about how foundation models empower providers to create tailored, adaptive solutions through smart data analysis. I'm convinced that cloud and AI together has the power to reshape healthcare more in the next decade than we have seen in the past century. But let's not forget, we are dealing with people's health. So we need to implement these technologies that are safe and making sure we are thoughtful and respectful, as well as responsible. So I'll spend most of the time today talking about frameworks for managing risk associated with generative AI, as well as these multimodal foundation models. Let's start at the very beginning. In a couple of years back with ChatGPT, which was one of the first technologies to showcase the incredible potential of generative AI. To understand ChatGPT, we break it down into 4 major components. The first is the transformer architecture. We won't go into the details of the architecture, but this architecture lets capabilities or components like ChatGPT to understand and generate human-like language by recognizing complex patterns, relationships, and context, as we saw in the previous session as well. Secondly, the advances in compute power has allowed us to process huge amounts of data in parallel. Thirdly, alignment, instruction, or human reinforcement learning shapes generative AI by using human feedback to refine models' output for accuracy, relevance, as well as looking into the alignment with human values. As we'll see today, having humans in the loop is essential for rolling these technologies out responsibly. Finally, we'll talk about foundation models, which are the backbone of generative AI, and that's where we'll focus next on. Foundation models at the core are trained on vast amounts of data, and that is often unlabeled, giving them a strong foundation to tackle a wide array of tasks without requiring significant data sets upfront. We are seeing these models make a huge impact where models like ChatGPT, Claude for language processing, as well as are also used in healthcare for a wide variety of medical imaging and multimodal tasks as well. These models started as language, but one of the key powers of these technologies is they were multimodal. They always started with multimodal. So that's what we have seen over the last 18 months. It started with language, now they're adding voice, they're adding images. We looked into the previous example, how easier it was to even add pathology into some of these components as well. And because these technologies are built on diverse data sets, they are incredibly adaptive. They can be fine-tuned for specific tasks, whether it's answering complex questions, generating unique content, or detecting conditions in medical images. To give an example, at G Healthcare as well, we have been doing research into foundation models in X-rays and MRs, which could be game changers. For instance, these foundation models could help analyze images faster, more accurately, generate automated reports that supports radiologists in their daily workload. Another major advantage of these technologies is that they can handle different kinds of data sets. It could be voice, it could be images, it could be text, both at the input level as well as at the output level. This versatility to handle multimodal data set at the input level as well as at the output level is kind of game-changing as well as invaluable, especially in complex field like healthcare. Next, talking about foundation models and how are they different than traditional AI models. Foundation models represent a quantum leap over traditional AI models in three key areas. Firstly, traditional AI models rely on limited and narrow data sets. Foundation models, however, are trained on vast data sets, capturing a wide range of knowledge. This results in actually more reliable and insightful predictions. Older models are often defined or designed for single tasks. It could be text-only or image-only processing. However, foundation models being multimodal can actually handle multiple types of data sets simultaneously. This capability opens up new possibilities for richer and more contextualized AI applications. Finally, traditional models are usually built for specific tasks and lack flexibility to adapt to new context. This can lead to longer development cycle as well as massively increased cost for the healthcare continuum. Foundation models, by contrast, bring broad adaptability, where they can be fine-tuned for various applications, making them more cost-effective and impactful across multiple domains. I'll give an example over here of how foundation models can transform breast cancer care pathway. Today, patients move through a complex workflow, from screening to diagnosis to treatment, as well as to the monitoring, where they're going through various devices and components, such as ultrasounds, biopsies, MRIs, and PET scans. Because of the challenges associated with traditional AI that we just talked about, we see various challenges that can arise. For instance, that can lead to inconsistent screening adoption, complex biopsies, as well as various other challenges. It can also lead to low trial enrollment, which would slow innovation, while it can also lead to treatment selection, guideline adherence, and follow-ups, which remains inconsistent. Foundation models being flexible, multimodal, and adaptable can actually help streamline this fragmented process, addressing these key pain points with Now let's bring all of this together and explore how these innovations can actually translate to tangible outcomes that can actually improve throughout the patient journey. Traditional machine learning traps data from each clinical modality in separate silos, which creates significant inefficiencies. Foundation models, however, break those barriers by synthesizing complex, multimodal healthcare, longitudinal healthcare data from diverse sources into a unified view. These models are highly adaptive. So over here, we can see an example, and we can actually think of an oncology-focused foundation model for breast cancer, for instance, can actually be adapted to other cancer-related areas like prostate cancer. They can even go across multiple care areas where a model from oncology can go into cardiology or neurology. Take Alzheimer's, for example. Imagine if a new drug receives regulatory approval. Foundation models could help develop the new care pathway solution in the matter of weeks versus months or years that it takes today, revolutionizing how quickly we can adapt advancements in the field of medicine. Next, we'll talk about some of the considerations and measurements that we need to ensure to make sure we can actually bring some of these GenAI-enabled devices into the system. To ensure the responsible rollout of these technologies, I'll focus on four main pillars. Firstly, we'll discuss about the defined intended use, which is critical. By clearly outlining the product, what the product is designed to do, we can actually set the foundation for every subsequent step, from design to regulatory compliance. This clarity ensures that product aligns with the user needs and industry standards. Secondly, adopting robust risk management practices is essential. By identifying and addressing potential issues early, we can enhance safety, improve reliability, and build trust with users and stakeholders. Pre-market evaluation comes next. This involves rigorous testing and validation before the product goes to the market, catching in on potential problems and ensuring it meets the quality and regulatory standards. And finally, establishing change control and lifecycle management is key. These systems allow us to manage updates and improvements efficiently, keeping the product compliant, high-performing, and relevant throughout its lifecycle. With these measures, we can navigate the transformative potential of generative AI responsibly and safely. Next, we'll go mainly discussing two major topics, which is first is the intended use and then around risk management. The concept of intended use is fundamental to the FDA regulatory system. For a medical device, its level of risk depends on its defined intended use. This principle also applies to GenAI-enabled products. To scope the intended use of a foundation model or a generative AI, developers can take several approaches. For example, they can control the prompts to shape the system's behavior. They can create boundaries on what model can generate. This allows developers to focus the product on specific use cases. Defining use cases or intended use case initially is going to be critical for ensuring the trust and effectiveness, starting with a specific intended use case and expanding it gradually. From the regulatory standpoint, the process starts with the first FDA authorization that corresponds to the scope of the initial use case, complemented by predetermined change control plan or PCCP that allows us to use these use cases to expand over time with the same intended use case. In this slide, we can see an example of how we might want to start with, for instance, cardiac segmentation in ultrasound using GenAI or foundation model tool, and later expand to segment structures in kidney or other organs. We can do this as the intended purpose and workflow remains unchanged. However, detecting and categorizing lesions might represent a new intended use case as it involves interpreting clinical significance and influencing diagnostic decisions as well. A similar process can actually be used even for GenAI or agentic AI systems designed to fulfill a specific task as we seek to expand the universe of possible use cases. Medical device manufacturers use established risk management methods by assessing the impact of failure modes in the context of use, as well as designing mitigations to address the possible hazards. The approach equally applies for GenAI-enabled medical devices as well. However, GenAI and foundation models also introduce new failure modes, like hallucinations, where systems can generate false or misleading information. The good news is, by anticipating these use cases, we can proactively design effective mitigations, ensuring safe and more reliable use of these advanced technologies. This structured approach helps systematically to identify as well as to address potential failures to enhance device safety and reliability. Just like we would do with medical devices, we have to be cognizant of the risk involved in the rollout of these technologies and think about strategies to address them. There are some of these components which are common for even traditional AI models. For instance, misrepresentation of information can happen even with traditional AI systems, and we have to be aware of these limitations. We can apply techniques such as explainability and transparency to address them. For GenAI systems, which can also produce hallucinations, ontology-based methods, or reasoning, something these models are really good at, can ensure accurate and reliable information generation. We can address the risk. One of the common questions comes around is the inconsistent output that these GenAI solutions can build out. We can actually de-risk this risk associated with the inconsistent output with mechanisms such as temperature control, which can help us to ensure that the model output remains consistent and repeatable over time. Finally, across the board, we find that issues can be mitigated by keeping human in the loop, allowing for essential oversight and judgment in critical decisions. The level of human involvement is dependent on the intended use case that we look across these different areas as well. Now let's look at an example of how we might mitigate risk. AI models trained mainly on common diseases may struggle to recognize rare conditions, leading to potential misdiagnosis. For instance, a patient with rare autoimmune disease might be misdiagnosed with a common condition like rheumatoid arthritis due to gaps in AI's training data. A machine reasoning algorithm can actually help by enabling the AI to use reasoning, which can be both deductive and inductive approaches, which can be enabled by chain of thought reasoning to better navigate complex medical cases. This can be extended with ontology-based disease models that can provide structured framework for organizing and sharing medical knowledge, which can improve the diagnosis, treatment, and research efficiency. So by bringing together machine reasoning and ontology-based models, we can actually enhance AI's capability to manage both the common as well as the rare conditions in healthcare. Now I want to give another example and another technique that can be used to mitigate risk. Visual grounding, which involves locating relevant objects in an image using natural language descriptions, enhancing image interpretation and diagnosis in medical context. This technology improves image understanding by allowing for more precise identification of abnormalities. In the example of this slide, a radiologist generates a report describing a condition observed in an X-ray, such as pneumothorax or lung consolidation. The report is processed by medical report grounding system, which highlights specific areas in X-ray that correspond to the descriptions, assisting the clinician by visually marking relevant regions and ensuring consistency in interpretation. One could imagine the same visual grounding system to also apply to the output of a foundation X-ray model, generating a report that links directly to the content in the image, kind of creating a flywheel around that as well, which kind of eases the review by the radiologist. This is just one of the many examples, and there are many more which we can't cover with a shortage of time, but we'll be covering more of these mitigation techniques in the docket that will be available in coming weeks as well. One way to leverage the power of AI is to select the foundation model that best aligns with your development needs, and then fine-tune it using domain-specific data for a specific task. For example, with SonoSam, we started with Meta's promptable image segmentation model, which was initially trained on 1 billion segmentation masks from natural images. We then further tuned this model on 200K more ultrasound image masks. In this case, we didn't know if SAM was trained on ultrasound images or not. So our first approach was to understand its baseline performance. That's when it was determined that fine-tuning would be essential in this use case. We eventually compared the performance of SAM and SonoSAM on an independent and representative dataset of ultrasound scans to understand the behavior of model on a variety of clinical images. It's important to note that the level of information about the foundation model or dataset used for pre-training does not significantly impact the process used for validation. We validated the fine-tuned model on an independent, representative, and diverse dataset against the ground truth to ensure adequate performance as outlined in this paper as well. As I near the end of my talk, I want to take you through three-step framework to think about change control and device management for GenAI-enabled medical devices. Firstly, change control, which is maintaining appropriate version control and end-to-end validation, ensures that GenAI-enabled devices produce consistent and reliable output. Adaptability and speed. We just saw that by leveraging PCCP or pre-configured control plan, we can safely expand the model's use case within its intended scope. This setup can also be used for quick iterations, such as adopting new foundation model versions to keep pace with advancements without compromising safety. And lastly, with post-model monitoring, by applying best software practices are essential for capturing feedback during real-world use. And we have seen some of them today, and we'll share more of those in future as well. So in conclusion, I hope you got the sense of how excited we are about foundation model generative AI and how they can have significant potential to transform healthcare. With the right controls, this can effectively be integrated into medical devices to enhance their functionality. Leveraging existing best technological practices within the current regulatory framework allows us to manage these integrations responsibly. With what's more, combining foundation models with PCCP enables us to implement changes safely and efficiently, ensuring timely advancements while maintaining high safety standards. Thank you for your attention today. Thanks for your time.
Thank you very much, Dr. Bhatia. Our last presenter for the morning is Dr. Pranav Rajpurkar, who is the Assistant Professor at Harvard University. Dr. Rajpurkar, please, your presentation. Thank you.
Delighted to be here today. Thank you so much for having me. My goal today is to share some of the lessons that we have learned in trying to evaluate generative AI applications. Here's my disclosure. There are many applications where we can have generative AI make big impact. One of them is the ability to generate text from images, be able to summarize clinical notes, be able to record conversations between patients and doctors, and to be able to summarize them. It's not just about text outputs. We can enhance images. We can generate visualizations, which can all be helpful to different stakeholders. A few months ago, I was shadowing a GI colleague of mine at one of the Boston hospitals, and they use these narrow AIs to be able to highlight colon polyps on the screen. There was a time at which one of these detections came, and the GI doc didn't know what it was about. Wouldn't it be cool if we would be able to ask an AI model a question, explain the object appearing on the screen, and the AI model could understand the intent and be able to answer that query in real time? These are the kind of technologies that I'll be talking about today. The reason that we're seeing a lot of advances in generative AI is because of technology advancement on three fronts. One of them is the ability to leverage unlabeled data. Another, the ability to leverage different kinds of data or multimodal data. And then the ability to communicate that in natural language. So the main question I want to talk about today is, if we have a generalist AI system that can generate these text outputs for a variety of inputs, then how do we think about evaluation? And I want to break it down into two fronts. One is, how do we think about automatic metrics, which allow us to do things at scale? And the second is, what is the role of humans in the loop, of experts in the loop, as part of this evaluation? So I want to start with the question of metrics. What metrics allow us to determine in this case, whether a report that's written by an AI matches a report that's been written by a human. Here are the examples that we can see on the left, and we're going to look at different metrics on the right. So we started working on this problem in 2020, and one of the literature fields which has looked at this question a lot has been the field of machine translation, where we have translation produced by a model and then a translation written by a human. And there, one of the most common metrics are metrics like BLEU. And what BLEU does is it looks at word overlap to determine similarity. For example, here there's a perfect match on certain terms like air bronchograms, and this is reliable when identical terms are used. However, this often fails where different words are used to convey the same meaning. So left lower and left base, consolidation opacity, effusion fluid collection. This is not something these can recognize. So how do we solve this? So at the time, we turned to thinking about embedding-based metrics. Embedding-based metrics decide how to embed every word such that we capture the meaning of that word. Here, consolidation and opacity would be considered similar, and we were using this metric called BERT score. Effusion and fluid collection now becomes similar. However, one pitfall of these metrics that often happens is that opposite meanings can be represented very similarly. So with and without mean two very different things. We want them to be far apart, yet these metrics often don't distinguish between the negation and the presence of something. So in 2020, we started to think, how do we design a metric that captures what matters in clinical medicine? And so we came up with this clinical accuracy metric called CheckSPERT to evaluate the medical context within the text rather than the text itself. So here, understanding things like with and without mean opposites, correctly mapping synonyms to standard terms, preserving the negation are all the positive things. However, this took us several months to build and was limited to a set of limited conditions. We also could not link these findings to the locations in which these findings were occurring. So if you have a left pleural effusion versus a right one, this is not something this metric would be able to help us disentangle. So how do we solve it? So we started to think, how can we extract medical knowledge from these generations? And we developed a metric called RADGRAPH F1, where the idea is each one of these reports contains medical context. Can we explicitly extract the medical knowledge here? And so we generate these graphs off of these two reports, like you see, which captures the different entities and the relationships of those entities to their absence or presence, but also to the anatomical location. And that's very helpful. Now we can compare these two graphs with respect to each other. The advantage of this is that we can capture a bunch of findings, we can preserve the relationship between the finding and the anatomy, and we can handle negation. But this is quite hard to extend across modalities. RADGRAPH F1 was developed for chest x-rays. We collected a bunch of human labels. We created a model that would be able to automate part of that. And then the second challenge is it's very hard to normalize across entities. To be able to understand that one medical term is actually a subset of another medical term is a challenge that's very hard to solve. So fast forward 3 years later, we have been working very actively on this front. A metric that we developed called HEAD-CT1 is able to compare two reports using knowledge ontologies. Here, the original report and an AI-generated report have both been tagged with the anatomies, the descriptors, and whether or not an observation is present or absent. So what you can see on the right is we can now understand that the extracted entity of brain and cerebral mean the same thing, atrophic changes and atrophy mean the same thing, and the small vessel disease changes means the same thing as microvascular ischemic changes. And we understand that the content of these two reports, where it matters, is the same. But I think one question that we should start asking when we think about automated metrics is, does this align with what we care about? In particular, if we asked experts to score this report, to say, this is the AI report, this is the generation, do you think this is correct? And they say there's one significant error here, there's one insignificant error here, do any of these metrics allow us to really get at that salient question? So how do we design this experiment? We thought in 2022, let's go ahead and understand the correlation between each one of these metrics and the expert rating. And so we have the radiologist score on the x-axis and the metric score on the y-axis here. And we're going to evaluate these 4 metrics. And here's what we found. We found that the expert scoring revealed the highest alignment with RADgraph F1 compared to BERT score, CheckSBERT, and Blue. But what we also found is that these different metrics are really capturing different ideas. And maybe it makes sense to think about how can we get the best of all worlds in creating a really good metric. And so we thought, could we compose these metrics? Could we take the weighted combination to be able to create the alignment with experts? And that's what we did with a metric that we proposed called RADClick, where we showed it has the highest correlation to experts and actually combines these different metrics together. But often, and certainly is going to be the case clinically, a single number that tells you how well a report matches another one is often not fine-grained enough to tell us what exactly is wrong here. And we believe there is a strong need to understand the source and the type of the errors in the generations are made. And we break it down into 2 questions. One is, what parts of the generations actually have errors? And 2, is this a significant error, or is this a stylistic one that won't make a difference to patient management? And so we developed this metric called FineRAD score. But before I talk about the metric, I want to talk about the idea here. The idea is, let's start with an AI-generated report, and let's ask experts to fix it line by line. Now, they might decide to remove certain lines, they might decide to edit certain lines, and they might decide to add certain lines. Each one of these edits has a cost associated with it, or a level of significance associated with it. For example, the first 2 errors might be non-actionable or actionable non-urgent errors, while we might have an emergent error. And we really care about catching those emergent errors. We don't want to make worst-case mistakes. Here's a comparison of what happens when you look at the average error made by an AI generation and a human. If you look at the different error severities on average, you can see that if we take a look at something that's a clinically significant but non-emergent error, which is at 2, 68% of AI reports and 69% of human reports have errors of 2 or less. And this seems great. It's almost equivalent. But it doesn't tell you and doesn't tell us the full story here, which is, if you don't look at the average error, you look at the maximum error. What is the worst error that is produced by the AI? You see that there is actually a significant difference between the AI generation and the human generation. And this goes to show and say that we should not be thinking about averages. We should also be thinking about worst-case analyses. One of the things that our lab is actively working on is, can we automate this process of fixing AI-generated reports, given access to the expert reports, such that it's not something we have to go out and seek experts for in order to be able to do, and FineRAD score performs this task pretty well. So I want to switch gears a little bit and talk about current state of the arts in terms of building these generalist models, and I want to share one work from our group called MedVersa. The idea of MedVersa is to think about building a model that goes across different modalities. Typically, we have different models that work on chest x-rays, on dermoscopy images, on CT images. Would it be that crazy to have a single model that could look at all these kinds of images and be able to perform a host of different tasks? This might be tasks like the ones we've talked about today, where we're generating a report, but it might also be traditional tasks like being able to classify a disease off of the image, or even being able to outline the area of interest, draw a bounding box. What we found was that when we trained a single model to be able to do these tasks, we found that MedVersa was able to outperform the previous state of the art. On medical report generation, we're here using RAD-click. On being able to classify images, we're using the F1 score. On being able to draw bounding boxes, we're using the intersection over union. And as we've seen earlier today, lots of images are used to be able to actually train such a model. And this idea that we're working towards generalist models that can actually perform better than the specialist models is fairly new, because it's been long assumed that if you want something that works really, really well for this specific task, we should only go ahead and train for that specific task. That no longer seems to be the case. So I want to hopefully show a demo here of this model. So I want to show here a demo of MedVersa as it goes through and generates a report on a chest X-ray image. You can see it outputs the output here for this image that should hopefully be visible now. One of the things we expect these models to do is look at the context. So here it's a 70 to 80-year-old female, and we're going to generate a report here. One of the findings is moderate pulmonary edema. We should be able to ask a question particularly on that finding. So to say, how severe is the edema? And it should say an answer that's consistent with the report that it's generated. Now, we might want to do a task like being able to outline the heart. It's not quite right here, but I like to say its heart's in the right place. We'll have to think about how to evaluate such systems, but it can look at dermoscopy images, be able to make a diagnosis, be able to perform tasks like segmentation on 3D modalities, here being used to segment out the liver, and having a new task be solved be as simple as simply changing the prompt used in this particular case. So that's MedVersa. And I wanted to share this example as an example of the capabilities that we're thinking about when we're talking about the next generation of multimodal generative AI models and what it takes for us to evaluate them. So I want to now talk about human-centered evaluation, because that's such a key area. And one thing I'll mention before we do that is, if we look at automated metrics, what I just showed you today, MedVersa, is currently the best benchmark model in terms of generating reports on chest X-rays. Over the course of the next year or two, I expect we'll see significant progress here, and benchmarks are going to be very important. And we have the RexRank benchmark that we have generated for this now. So on to human evaluation. How do you leverage expert capabilities to be able to evaluate such a generalist system? Well, one of the ways that we thought about it was to say, we're going to ask experts to choose which report is better. One of them is going to be generated by another expert, one of them by the AI model. Now, the expert might say both of them are equivalent clinically, or might say report A is better or report B is better. And what we found was that overall, expert-written reports are preferred. AI-written reports are preferred some of the time, and they're equivalent more than 50% of the time. We have a breakdown of how things perform on abnormal cases as well as normal cases. And on abnormal, you see the human preference being greater. And for normal, you see they're pretty much equivalent most of the time, and an equal split of the remainder between whether the AI was preferred or the human was preferred. One of the other ways in which we think about evaluation is to think about what is the setup in which they're going to be used in practice. One of the exciting opportunities here for radiologists is to be able to spend less time on cases. The idea of working off of a draft and being able to edit it to sign off is not new. That's how academic radiology often works. And so could we have a system in which these AI drafts are given to experts for them to modify and then sign off? The way we set this up is we say we're going to measure the time spent on a study, the mental effort, and the confidence in each report as radiologists are going through X-rays. The control here is going to be starting off with a normal negative template, and the AI model as well as the radiologist is going to be supplied with the demographic, the indication, and also the comparison. And what we found here in a small pilot study right now is that there is, in fact, significant time savings and mental effort reduction, as well as increased confidence in each study. But the caveat here is that it's going to be very important to think about how this technology can work for everyone. Even in small-scale trials, we're seeing some people experience big benefits from this technology, while others show no improvement in their performance or their time taken as a result of these technologies. I want to switch gears a little bit and talk about one of the things that we've all been very excited about, is the ability of large language models to be able to have conversations. Now, there's a big disconnect in the way that these systems are evaluated and the ways in which these systems are expected to be used in practice. And I want to share one example of that. The primary evaluation for LLMs right now is based on answering medical exam questions. And we've seen incredible numbers in terms of the ability of LLMs to outperform humans on this task, several studies, including the ones this week, that even show that the AI alone performed better than the clinician being assisted by the AI. But these exam questions and the setup is broken in fundamental ways. One is that no patient ever presents with, here's a summary of my symptoms, here's my vignette. No patient talks about their medical terminology in such precise terms. And no patient gives options for a doctor to choose between. And these are ways in which we need to change the way we're thinking about the evaluation of these clinical large language models. One of the ways in which we are thinking about this is how can we have benchmarks for conversational assessment of LLMs. Now, we can get these LLMs to have conversations with lots of patients, but that would be unethical in many ways. Could we have LLMs simulate patients to have these conversations with clinical LLMs and see in that setting whether or not these clinical LLMs do well, is how we're thinking about this. So I want to present our results here on evaluating clinical LLMs on 12 medical specialties, both text-only and multimodal LLMs, and talk a little bit about continuous monitoring here. The setups we have is we have these case vignette evaluations, which we know are unrealistic. And what we do is we take these case vignette evaluations and ask a patient to share information with the clinical LLM. The clinical LLM asks questions to the simulated patient. The patient answers using the available information in the vignette. And we evaluate this in a multi-turn, in a single turn, and a summarized conversation setting. The main point was this, that when we actually get these clinical LLMs to have conversations, to ask the right questions, they actually do much, much more poorly than when they're given the vignette. A large part of this has to do with incomplete gathering of relevant medical history for these clinical LLMs to actually have all the information to arrive at the right diagnosis. Similarly, multimodal LLMs are severely limited in their image interpretation capabilities, and this becomes worse when we try to evaluate them in this conversational setup. I want to end with talking about an application of generative AI that has several different components, but goes to show the potential value in terms of helping patients understand their own medical information. A lot of us, or our family members, might have had experiences in which we get scans and we get radiology reports associated with those scans. And the scans come on CDs and the reports come over email. And it's very hard to understand what any line of the report means, especially for those who might not be medically trained. So we thought about, could we solve this problem by creating a system which allows us to understand different lines in the salient information in the radiology report, and in particular be able to link it to regions of the image and also regions of the anatomy. This would allow a patient or their family member to understand what's going on in their image, what was found, what does it look like, be able to compare to what a normal scan looks like, be able to see the organ, and also have this virtual avatar do some explaining. Now, this is a system that can be built with a combination of existing components and components which we developed to segment out abnormalities. And I want to play you a demo of what this system looks like. Okay, so in this particular setting, there's no audio, so I'm going to be the virtual avatar in this case. But the first finding that's being explained is that of aortic calcifications. And as the system scrolls through the aorta, you can go ahead and have the virtual avatar talk about these. We're not going to go through the whole video, but if we fast forward it, we'd also have a comparison to the reference normal image right there, and then also the organ rendering on the right. And this is our vision for what might be a world in which we have the patient be empowered to make their own medical decisions. We can go back to the slides. So in conclusion today, I wanted to share some of our lessons of evaluating LLMs and generative AI. And what I hope to have shared with you is that it's challenging, is that there are automated metrics that take us a long way, that there are human-centered evaluations that take us a long way as well. The space of opportunities is immense, and it's a tremendously potentially helpful technology, and we need to think about the best way and the most safe way of bringing it to practice. I'd like to thank my lab members who made all this possible. Thank you.